Greetings. My name is Howard Levine, and I'm a NASA project scientist at Kennedy Space Center. This is a video for a presentation entitled Design and Ground Studies with the Passive Orbital Nutrient Delivery System, PONDS. My co-authors are Jeffrey Richards, Larry Koss, Ye Zhang, Anna Nguyen, Mark Weislogel, and David Reed. Long-duration space missions will require a fresh food supply to supplement crew diets, which means growing plants in space. The Vegetable Production System, or Veggie, was developed to be a simple, high-growth volume facility capable of producing fresh vegetables on ISS. Veggie contains an adjustable light-emitting diode light bank and uses plant pillows with quick disconnect fittings for the addition of water. The pillows are filled with an arsenite substrate containing slow-release fertilizer pellets for nutrient provision, and together the system has successfully been used to produce a variety of salad crop species for both crew consumption and research purposes. At Kennedy Space Center, we have developed another approach for growing plants within the veggie called PONDS. PONDS uses a plant cylinder filled with the arsenite fertilizer pellet mixture that is inserted into an exterior reservoir to which water is added. PONDS contains oxygen diffusible membranes both on the plant cylinder itself and on the outside of the reservoir section. Using our PONDS prototypes, we successfully grew a variety of plant species as seen below. Here are results from an outrageous lettuce test in an early PONDS unit. The picture shows lettuce status at days 4, 6, 12, 20, and 39. 3 seeds were planted in each PONDS unit and thinned to 1 on day 6. Here, and in all of the following growth studies, the growth chamber conditions were 85% red to 15% blue LEDs, light intensities of 200 to 250 micromoles per meter squared second, temperature of 22 degrees centigrade, a light-dark cycle of 16 to 8, a CO2 set point of 2,000 parts per million, and a relative humidity set point of 40 to 45%. The lettuce plant was harvested for the first time on day 39 and yielded 18 leaves with a fresh weight of 35.6 grams. Below you can see how the plant looked immediately after this harvest and how quickly the leaves grew back by 4 and 12 days after harvest. The second harvest occurred 22 days after the first harvest and yielded 18 lettuce leaves with a fresh weight of 28.8 grams. Below you can see how the plant looked immediately after the second harvest and when the flowering stalk first appeared and subsequently developed. After the second harvest, there was also a change in the phenotype of the lettuce leaves with a much more velvety morphology appearing. Here are some pictures of the subsequent flower and seed development that occurred. Seed formation demonstrated self-pollination, and 95% seed germination demonstrated self-compatibility. Here are the results from a red robin tomato experiment undertaken in one of the KSC PONDS prototype units. Flowers started opening on day 34. Fruit formation was observed on day 41, and ripe tomatoes obtained by day 86. The first harvest occurred on day 92. A second harvest occurred on day 185, and a third harvest occurred on day 283. We were able to keep these plants growing by adding Miracle-Gro nutrient supplements at dilutions of 1 to 1,000 into the exterior reservoir starting at the time of flower initiation. These were initially performed once a week and progressed up to the 3 times a week procedure. We were able to track water usage by way of a window on the side of the ponds unit that was covered between observations. Cumulative water usage is shown with the solid black line, and daily water usage shown with the red squares. You can see drastic water usage drops immediately after harvests 1 and 2, when the leaves were significantly cut back. Minimal leaf formation and possibly a general senescence of the plant after the second harvest resulted in low water usage rates during this interval. As was done on ISS using the veggie pillow method, we demonstrated excellent zinnia flower formation within a KSE ponds prototype unit. Again, we were able to get prolonged growth and flower formation for close to a year using the Miracle-Gro nutrient supplementation procedure as was used for the tomato experiment. And of course, for basic research experiments with Arabidopsis thaliana, we demonstrated the ability of ponds to support excellent Arabidopsis growth. We then partnered with the TechShot and Tupperware corporations to take the design to flight-rated versions using Tupperware's injection molding technology. Here you can see an early version of the flight-rated ponds showing how 6 of the units can be used with a single veggie. In the bottom left figure, you can see a cross-section of a ponds unit showing a wicking material extending from the exterior reservoir section up through the plant cylinder to the point where the seeds are attached for on-orbit imbibition and germination. The bottom right figure is an exploded view showing quick disconnects into both the exterior reservoir and the plant cylinder for water additions. Also visible is a clear germination cap to maintain high humidity conditions for seed germination. Once the seedlings are established, it is removed. This slide shows some of the details of the flight version of ponds. In A, you can see the water input tube, which intentionally introduces water to the base of the plant cylinder. In B, you can see the quick disconnect that is used to introduce water directly into the plant cylinder at the start of the experiment. Diagram C shows capillary mat fins extending outward from the plant cylinder, and D is a top view showing capillary mat fins extending outward from the plant cylinder. Diagram E shows the approximate pond's water holding capacity, which in the plant cylinder is 115 milliliters, and the exterior reservoir 400 milliliters, for a total of 515 milliliters. Diagrams F and G are views of transparent ponds units without the covers used to block light entry. We performed a high-fidelity science verification test using the flight-rated ponds units at Kennedy Space Center. 3 lettuce seeds were planted in each ponds unit. On day 0, 120 milliliters was added to the plant cylinder to initiate germination. On day 5, 100 milliliters was added to the exterior reservoir to prevent the drying out of the seeds. On day 8, the germination caps were removed, seedlings were thinned to 1 per ponds unit, and another 100 milliliters added to the exterior reservoir. Additional additions of 200 milliliters to the exterior reservoirs were made on days 15, 23, and 27. In the pictures below, you can see that by day 5, 14 of the 16 planted seeds had germinated. On top, you can see the day 8 lettuce pictures after seedling thinning down to 1 seedling per ponds unit. And below, you can see the day 12 lettuce pictures. Here you can see the lettuce growth progression by day 15 on top, and on the lower left, you can see the pond's exterior reservoir on day 15 before the addition of 200 milliliters. And then on the right, after the addition of 200 milliliters. Here are the day 19 lettuce pictures. This slide shows the final results of the 29-day test conducted at Kennedy Space Center with the flight-rated version of PONDS. The picture on the left is a top view of the lettuce from 6 PONDS units that were situated within a single veggie unit. On the top right, you can see a lettuce plant from a single PONDS unit, and on the bottom right, you can see roots that have grown outward from the plant cylinder into the exterior reservoir. This graph shows the evapotranspiration data from all 6 PONDS units. Minimal water loss occurred until day 8 when the germination caps were removed. Maximum PONDS water loss topped out at 66 milliliters per day. Later this year, we hope to validate the operation of PONDS under microgravity conditions on the International Space Station. Wish us luck. Many thanks to members of Kennedy Space Center's Utilization and Life Sciences Office and the NASA Biological and Physical Sciences Division for support of the PONDS development project. Thank you for watching.